so let's begin. Now, here's the first thing that you have in your book, the first aid. I usually make or prepare this lesson based on the first aid topics. That means whatever there is in first aid, the same content I follow. Now, here's the first thing that is discussed in the first aid in the endocrine section is the embryology of the thyroid gland. Now, how the thyroid gland is formed. Now, as you can see in this developing fetus, at the side view of this developing fetus, you can see that usually we are like when we are in our mother's womb, we're like this, right? This kind of structure. Now, this view is looking from the side. This view. Now, as you can see that in the um, area here that is called in the uh, gut area or in the foregut area, usually in the tongue, the base of the tongue, there's a protrusion that occurs here that is called the thyroid primordium, or sometimes this thing is called the primordial thyroid. Now, this is a protrusion that goes down or that descends, and usually it forms into the thyroid gland. So it's a protrusion from the base of the tongue where this thyroid goes all the way down into where the thyroid is located in our neck, right? Now, so this is a endoderm derivative. This thyroid is mostly endoderm derivative, but there are some neural crest derivatives of the thyroid gland. We know that when you go to the histology section, you'll see that some of the portion, the follicular cell of the thyroid is derived from the endoderm, and the parafollicular cell or the C cell, those are derived from the neural crest cell. Right, so the next picture we see the descent of thyroid. That means how from the base of the tongue this thing descends down. This thyroid, it follows a path. And as you can see, there's this hyoid bone. It crosses the hyoid bone in front. It goes below down the thyroid cartilage, and it sits here, the thyroid gland just under the thyroid cartilage. Now, there's this when this thing descends down, this thyroid descends down, it has a duct that connects the base of the tongue with the thyroid. And this duct is called the thyroglossal duct. This is the thyroglossal duct. Usually it disappears, but sometimes it happens that it may remain as a remnant, or it may form into a cyst. It may convert into a cyst. Or sometimes you find it as middle lobe or a pyramidal lobe of thyroid. It means this whole duct thing, this thyroglossal ducting usually disappears. But if it remains, this thyroglossal duct remains. If you look at the thyroid, let's say the normal thyroids are like this, right? It looks like a bat to me. And sometimes it happens that it might remain as this duct, remnant of this duct may remain as a protrusion. We call it pyramidal lobe of the thyroid, or sometimes the whole cyst, the thyroglossal duct, um, may remain as a remnant, or it does not disappear, and there can be fluid collection, and that can lead to formation of the thyroglossal duct cyst. Now, the base of the tongue, if you look at the base of the tongue, and I hope you know that in our anatomy discussion in our early days of med school, you already learned that in the base of the tongue, there's a foramen. We call it foramen cecum. And usually that is the remnant, or usually normal remnant of this thyroglossal duct, or you can say the beginning of the descent of the thyroid. When the thyroid descends down through this path, this foramen remains as a remnant here. Now, this thing is called foramen cecum. All right. Now, next picture, you see this thing is the total gross view of the thyroid from the front. As you can see, this is the thyroid cartilage, and here is a thyroglossal duct. Usually it disappears, but if it does not, what happens that fluid collects here, and it may appear as a cyst. A cyst is a abnormal collection of fluid surrounded by a membrane, and as this thyroglossal duct can enlarge and can appear as cyst, your thyroid gland is here, and thymus in the very down part of this picture. Thymus is the place where the T cell, or the immune T lymphocyte cell, that gets educated, right? Now, here is a imaging of the thyroglossal duct cyst, and here you can see a fluid-filled space that can be shown in the front of the neck. As you can see, understand that this is the vertebra, right, cervical vertebra, and here is the bone, the whitish one. This thing is a bone. In front of it, you may suspect, okay, it's a midline neck mass that is your thyroglossal duct cyst. Now, when there is thyroglossal duct cyst, there can be other cystic dilation in the neck area that may confuse you whether it is a thyroglossal duct cyst or whether it's something else. Now, how can you be sure about that? Now, another um, possibility of this thyroglossal duct cyst, or another possibility that you get a neck mass, is persistent cervical sinus. Now, before going to the persistent cervical sinus, you need to have the idea about the cervical sinus. What is that? Now, again, that picture that I told earlier about the embryo, that these embryos are like this, right? I mean, I don't remember, but I know that I was like this someday when I was inside my mother's womb. Now, here you see that this structure, this side view of the structure, you see this kind of shallow grooves, right? Now, if you cut here the embryo, you cut the embryo here, and you look from the front, you get a view like this, something like this. And I hope all of you are familiar with this view of the pharyngeal and branchial arches, pouches, and the pharyngeal arch, the pouch, and what is that? Pharyngeal arches, the pouches, this kind of clefts, this kind of thing, this view is kind of familiar to all of you. Now, this view, this determines different types of pharyngeal clefts. Now, as you can see, here are the clefts on the side view, that number 1, 2, 3, 4, and this 2, 3, 4 clefts, that means this number 2, 3, and 4, these clefts on the side, these grooves, they fuse together and they form a sinus or space, a blank or blind pouch-like space. This is called the cervical sinus. Now, this cervical sinus, if it becomes a cystic dilation, that means some fluid accumulates here and see it appears as a cyst, we call it persistent cervical sinus cyst. All right. Now, this cervical sinus cyst, persistent cervical sinus cyst, it can be a differential diagnosis of your thyroglossal duct cyst. But how can you differentiate between these two, whether it's a cervical sinus or whether it's a thyroglossal duct cyst? Now, usually this thyroglossal duct cyst, it's in the midline, and this cervical sinus cyst or persistent cervical sinus, it is on the lateral side of the neck. Another thing is that the thyroglossal duct cyst, this thing is usually connected with the tongue. So when you protrude the tongue or when you try to swallow something, there will be movement of that cyst. That means thyroglossal duct cyst will move with the protrusion or movement of the tongue. But it will not happen in this case if it is a lateral cervical sinus cyst or persistent cervical sinus. These are some differentiating point between these two. That means if it is a persistent cervical sinus cyst, then it will not move with the protrusion of the tongue or movement of the tongue or swallowing action. But in case of thyroglossal duct cyst, it will move. All right, now this is the histological picture of the thyroid follicular and parafollicular cell. The thyroid follicular cells are derived from endoderm, and the thyroid parafollicular cell, these are derived from neural crest cell. Now, as you can see that this is a thyroid histological picture of your thyroid, and this is a zoomed in picture, and these cells are your follicular cell, and surrounding the follicular cell or parallel to this follicular cell is your parafollicular cell. Now, this follicular cell, they are derived from the endoderm, and the parafollicular cell, they are from the neural crest cell. There's a neural crest derivative. Now, we learn or we read neural crest cell every time, but what is this neural crest cell? As you can see here, this is the origination point of the neural crest cell. That if you remember that in your embryology knowledge or idea, that when we're growing as embryo, some stage of our development, there was a two-layered embryo. That means the embryo was like this, right? Two-layered embryo. And then eventually it will convert into three-disc layer. We call it three-disc layer or trilaminar disc. The top one is the ectoderm, the middle one is the mesoderm, the bottom one is the endoderm, right? Now this ectoderm, the uh, superior surface of the ectoderm, underneath the ectoderm, there's a tube that grows. Underneath the ectoderm, if we look at the ectoderm from the front, you'll see a tube grows in just underneath the ectoderm. And this tube eventually converts into a solid tube. It is not just a tube, it converts into a solid tube. This is called the notochord. And this notochord releases some chemical mediators. What happens that the area of the ectoderm that is just above that tube, let's say this is the notochord, and the area of the ectoderm that is just above the notochord, that converts into something called neural ectoderm or neural ectoderm. Now, this neural ectoderm, what happens that there's a folding there. Let's say this is the neural ectoderm. There's a folding like this. There will be a folding like this. And eventually this folding converts into a tube that is called the neural tube. And while this neural tube is getting formed, we get our neural crest cell. Like here you can see this is the notochord, and on top of the notochord you have this neural ectoderm, and eventually this side borders of the neural ectoderm is kind of rolling and they are forming into the neural tube. And there is this edges of this folding, they kind of gets free, and this free cells, they are called the neural crest cell. And these cells are migratory cells. That means they can go in several places. And that's why you get this neural crest cell in your thyroid gland. And that's why you get your neural crest cell in the intestine. That's why you get your neural crest cell derivative in the heart. So they can migrate into places. So that is the neural crest cell, and you need to remember that the parafollicular cell, which produces the calcitonin, a calcium regulating hormone that is getting produced from the thyroid gland, this thing is made up, or this thing is originated from the neural crest cell. All right. Now, a little bit of discussion of thyroid anatomy. What is usually asked about that thyroid gland? What are the blood supply to your thyroid gland? And this type of simple discussion and some extra information that I wrote here. By the way, I sent this uh, hand out to my regular students, but also those who are new, you are joining here, or maybe you are not, you have registered but have not joined. I will send you this note, this handout to you after the class, because I need to have the email list, what I'll get after the class. So here you see that the superior thyroid artery and inferior thyroid artery, these are the two arteries that are supplying your thyroid gland. Here's the superior thyroid artery, here's the inferior thyroid artery. Sometimes they ask you what are the branches, what are the arteries these arteries are originated from. The superior thyroid artery is originated from the external carotid. The inferior thyroid artery is originated from the thyrocervical trunk, which is a branch of subclavian trunk. Now, also you see that superior thyroid artery vein, an external branch of superior laryngeal nerve that courses together in the neurovascular triad. That means there's a sheath of membrane that surrounds this three structure and form this triad. Now, sometimes it happens when we do the thyroidectomy operation, there's a chance of rupture of this surrounding structures, like this superior thyroid artery can be ruptured, superior thyroid vein can be ruptured, the laryngeal nerve can be ruptured, and this laryngeal nerve disruption or destruction will lead to lesion in the vocal cord, because this laryngeal nerve supplies the vocal cords also, and that can lead to worseness of the voice. All right. Now, the next thing is the anatomy of the adrenal gland. The adrenal means what? Adjacent to the renal. That means adjacent to the kidney. This gland is adjacent to the kidney, and that's why this is called the adrenal gland. Now, this adrenal gland, on anatomical discussion, this is divided into the outer part or the cortex and the inner part or the medulla. Now, from this different location of this adrenal gland, that is the outer part and the inner part, you're getting different hormones, and those hormones have different regulators. That means different regulators work on these different parts of this adrenal glands, and these hormones are getting secreted. Right, as you can see that histologically, this adrenal cortex is divided into three areas, and also functionally, these divisions are true. The outer layer is the zona glomerulosa. The cells of the zona glomerulosa are rounded or arched clusters of cell. That's the description of the cell that we get in the zona glomerulosa. And primarily, angiotensin 2 is the regulator of the secretion of hormones from the zona glomerulosa. The hormone that is secreted from the zona glomerulosa is our favorite mineralocorticoid, aldosterone, which helped in the blood pressure regulation, sodium and water uptake, and potassium and H plus excretion in the kidney. Zona fasciculata, this thing is the middle layer of the adrenal cortex. It is regulated by ACTH and CRAs. ACTH is the hormone that we get from the anterior pituitary, and the CRH is a hormone that is getting generated from the hypothalamus. ACTH stands for adrenocorticotropic hormone, and CRH stands for corticotropin releasing hormone. That means the CRH from the hypothalamus works on the anterior pituitary to release the ACTH. And this guy work on your zona fasciculata to release the glucocorticoid cortisol, which helps in regulation of the glucose and which helps in regulation of several things, several metabolism of our body. Zona reticularis, here you can see that ACTH and CRH is the primary regulator again, and androgens are the hormone that is produced from this layer, and the primary androgen is this DHEA, dihydroepiandrosterone, that we get from zona reticularis. In the medulla, there are chromaffin cells, and these chromaffin cells are regulated by our autonomic nervous system, more specifically the preganglionic sympathetic fiber, and from there we get the catecholamines, epinephrine and norepinephrine. Now here's a description of the cells of this different layers of your adrenal gland, and sometimes the questions are asked about what type of cell is this, or maybe they describe a histological criteria of the cell, and they ask that what type of hormone is getting produced from the cell. Like the zona glomerulosa, the cells are rounded or arched clusters of cell. Zona fasciculata, foamy appearing cells in column, and zona reticularis are basophilic cells, that is bluish cells in anastomosing cores. They have connection in between. Now this whole three layers, they are producing the different adrenal gland hormones that we get. Now, here's the regulation of the pituitary and the pituitary hormones. Now, as you can see in this picture, this is the anterior pituitary, this is the posterior pituitary. First picture, it is showing the hormones that we get from the posterior pituitary, the oxytocin and the ADH. The oxytocin is it causes the contraction of the uterus. It helps us get milk from our mother. Now, this ADH, also called the vasopressin, it has two major action. One is the vasopressin, that means it presses the vessel, that means squeeze the vessel. Also, it leads to antidiuretic hormone, that is, it stops or inhibits the diuresis. Now, these two hormones are regulated from the hypothalamus. From the hypothalamus, actually not regulated. These hormones are generated in the hypothalamus, and from the hypothalamus, through some pathway, these hormones are stored in the posterior pituitary. That is one thing you should keep in mind, that these posterior pituitary hormones are not generated in the posterior pituitary. Rather, they are generated in the hypothalamus, and they are brought down in the posterior pituitary, where they are stored, and whenever they are necessary, they are released from the posterior pituitary. Now, this other thing, the anterior pituitary, you can see that hypothalamus releases some hormone, and due to the action of those hormones, the anterior pituitary cell. Now, directly, the anterior pituitary cells are producing some hormone. Those are released from the anterior pituitary. So this is one of the functional difference between the anterior and posterior pituitary. The anterior pituitary produces hormone by itself, but it is regulated by hypothalamus. But the posterior pituitary, it cannot produce any hormone. It only stores the hormone that is produced in the hypothalamus. That means in the hypothalamus, these hormones are produced. Those are those hormones go down in this posterior pituitary. They are stored there, and from there, these hormones are released. Now, what are the hormones that we get in the anterior pituitary? We all know it, but here's some example like TSH, ACTH, FSH, and LH, growth hormone, prolactin, endorphins. These are the hormones that we get in the anterior pituitary and posterior pituitary. We discussed ADH and oxytocin. One thing is important that developmentally, this anterior and posterior pituitary are different. The anterior pituitary, it originates from something called the Redcase pouch. Anterior pituitary originates from something called the Redcase pouch, and that's a elevation of our oral ectoderm. That means the top of the floor of the mouth, the roof of the mouth in the growing embryo, the roof of the mouth, from there, there's elevation that forms a pouch-like structure. If you can imagine that if here's the embryo, let's say here's this embryo is lying like this, and this is the floor of the mouth, and this is the roof of the mouth, right? Now, from this roof of the mouth or oral ectoderm, you can see that there's a outpouching, and this outpouching is mixing up with another outpouching that is growing from the top part of the neural ectoderm. Now, these two pouches joins together and form the total pituitary gland. The anterior pituitary is coming from this roof of the mouth protrusion, and this protrusion is called the red case pouch, and the posterior pituitary is coming from the neural ectoderm. All right. So that is one of the important aspects of the embryological development of your pituitary gland. Now, these hormones of the pituitary gland, not a big deal. The anterior pituitary, also called the adenohypophysis. Adeno means hormone-producing cell or secretory cell. Now, as anterior pituitary generates hormone and it releases the hormone, so it is called adenohypophysis. But again, the posterior pituitary does not produce the hormone. It only stores the hormone that are produced in the hypothalamic nuclei. Now, anterior pituitary, as you can see, it secretes FSH, LH, ACTH, TSH, prolactin, and so on, these hormones. And one thing you should remember, this hormone has two units. One is the alpha subunit, one is the beta subunit. And what is found that the alpha subunit of these hormones are common to all. That means this TSH, LH, FSH, HCG, although HCG is not secreted from the pituitary gland, it is secreted from the placenta. But when you look at the structure or the chemical structure of this hormone, you'll find that the alpha subunit of these hormones are same thing, right? But the beta subunit, which actually differs this hormone from one another in case of functionality. And now, you can see that ACTH, MSH, and beta endorphin, these three hormones, they are derivative of some chemical that is called propionmelanocortin, and this is the reason actually why we get hyperpigmentation when we have this disease, the Addison disease. What happens in Addison disease? Although it's a discussion of the pathology, just to tell you that in Addison disease, the adrenal gland has insufficiency. That means it is not generating hormone. Now, when the adrenal gland is not generating hormone, usually what happens, that adrenal gland produces hormone, that hormone goes to the top in the pituitary, and it gives a negative feedback in the pituitary so that the ACTH is not released. Now, if the adrenal gland is non-functioning, the ACTH will continue to release because there is no negative feedback. Now, this lack of this adrenal gland that leads to increase in ACTH production also will increase in MSH production. And this MSH stands for melanocyte stimulating hormone. Now, this thing, what it does, it goes to the melanosome or melanocyte in your deeper layer of the epidermis, and where it stimulates those melanocytes to produce melanin, and that leads to hyperpigmentation. And that's why we get hyperpigmentation in this disease, the Addison's disease. Posterior pituitary, also called the neurohypophysis, and this thing, as you can see, stores and releases the vasopressin and oxytocin, which are not produced in the posterior pituitary, which is just its storage house. And this thing is this hormones are produced in the hypothalamus, which is just the structure above the pituitary in the brain. And this thing in the hypothalamus, there's some nuclei, that means collection of cell bodies in the central nervous system are called nuclei. And this nucleus is supraoptic and paraventricular nuclei, particularly because there are other nucleus in the hypothalamus. This two nucleus produces this hormone, and from this hypothalamus, these hormones are transported down to the posterior pituitary, and from the posterior pituitary, these hormones are released. All right, so now the endocrine pancreas, some work about this endocrine pancreas, the islets of Langerhans. We know that it's a collection of alpha, beta, and delta endocrine cell, and this different cell produces different hormones, and alpha produces glucagon, and they are usually found peripherally. As you can see, this oxyntic or pink color cell are the alpha cell. The beta cell, they produce insulin, and most of the cells are these beta cells, and the cells are beta cells, and there are some interspersed cell, those are called the gamma cell, and this gamma cell produces somatostatin, which is a inhibitor of growth hormone. Now, that's the overall discussion of the embryology and the anatomy of the endocrine system. Now we'll go to the discussion of the physiology. But before going there, let's take a break for 5 minutes. Then we'll come back and start the physiology, which is actually the most important part of the endocrine system. All right. So let's take a break for 5 minutes. We're coming back. And let's begin. The first thing that is important, the important hormone of our body, the insulin and the structure of the insulin that is shown here. Now, as you can see, this insulin consists of two chains. One is the alpha chain, one is the beta chain, and it's a peptide hormone. And as you see, that these chains are joined together by some disulfide bonds. But also you see that another extra layer of peptide or extra bundle of protein is here. This thing is called a C peptide, and this C peptide is usually when the insulin is not functional. Usually when the insulin is getting produced, this C peptide is a extra peptide that attaches with the insulin molecule. Eventually, when the insulin goes into functionality, the C peptide is detached. Now, this type of insulins are called a pro-insulin, insulin with C peptide. Now here is some discussion about the insulin. As we know that it's a protein hormone, so when the insulin is getting produced, it will follow the path of protein synthesis, right? First, the DNA for the insulin, it will be transcripted to some mRNA. That's the normal genetics. Then from this mRNA, we'll get the translation procedure and we'll get this protein, the insulin. Now, when this insulin is produced with the help of some ribosome, and we know that insulin is a secretory protein, so we know that basically the secretory proteins are generated in the ribosome. Those are attached to our endoplasmic reticulum. We have two types of endoplasmic reticulum in our cell. One is a smooth endoplasmic reticulum, another is the rough endoplasmic reticulum. And this endoplasmic reticulum, this rough one, they are coated with these ribosomes. Now, this insulin, while it is getting generated here, let's say insulin is getting generated in the ribosome that is attached to some endoplasmic reticulum, one signaling molecule attaches with this insulin. And that signaling molecule, what it does, it will pull this insulin inside the rough endoplasmic reticulum. All right. Now, this insulin, while it is generated and it has this signaling molecule attached to it, the signaling molecule, what is actually doing, it is signaling the insulin to go inside the rough endoplasmic reticulum. Because there is some other procedure that will happen inside this rough endoplasmic reticulum. Now, that insulin, which is yet outside of the rough endoplasmic reticulum, this thing is called the pre-proinsulin. And this pre-proinsulin eventually goes inside the rough endoplasmic reticulum along with the signaling molecule. And inside the rough endoplasmic reticulum, that signaling molecule will be cleaved, and as it is written here, cleavage of the pre-signal or signaling molecule. Then when this signaling molecule is cleaved, this thing will be called proinsulin, and this proinsulin is the ultimate insulin that will come out of your rough endoplasmic reticulum, go to the Golgi bodies, and eventually through the Golgi bodies, it will be released out of the cell when it is necessary. And I hope you know that insulin is getting generated and secreted from the beta cell of the pancreas. Now, while the insulin is getting secreted, the insulin and the C-peptide, both of them actually gets secreted, but insulin is the ultimate functional unit. C-peptide is just there, I think, to protect it from some disruption or destruction. This C-peptide is usually attached to the insulin. Now, if there is some pathology where we get increase in insulin synthesis, like there's a pathology of the beta cell increases in size and it produces a huge amount of insulin, we call it insulinoma. Now, in case of insulinoma, you'll get the insulin, that means the proinsulin, the active form of the insulin in the blood, because there is a huge amount of insulin getting generated, and also in there's some other in insulinoma you will also get C-peptide, because in insulinoma what happens, the beta cell has this tumorigenic change that leads to huge amount of insulin production, and as we know that insulin is getting generated along with C-peptide, so both of this thing, the proinsulin and the C-peptide, both of them will be elevated, the level of both of this thing will be elevated in case of insulinoma. There's some other thing like sulfonylurea is the medication. We call them insulin secretagogue. What does it mean? It means they stimulate the beta cell to secrete the insulin. Now, this sulfonylurea, when it stimulates the beta cell to secrete insulin, there will be release of insulin along with the C-peptide. But sometimes in your question, you might find this type of scenario where you suspect someone to be abusing the insulin medication. Now, this medication, if someone abused this insulin medication, that means someone takes the insulin exogenously. Usually, what we have, the insulin in the market or in the pharmacy, the insulin we have, those are synthesized insulin. Those are those are synthetic insulin. That means those insulins are not generated in a person's body. Rather than with the gene technology, we can generate a huge amount of insulin as commercial basis. Now, those insulin usually lacks the C-peptide. So if someone has increased level of insulin in their body, you can suspect whether it is an insulinoma, whether it's due to sulfonylurea use, or maybe it is exogenous insulin abuse. So how can you differentiate? To differentiate between the exogenous or endogenous insulin, you look for C-peptide. If the C-peptide is also elevated, you suspect it's an endogenous increased insulin production. But if you cannot find the C-peptide in that person's blood, you suspect this is exogenous insulin abuse. All right. Now here in this picture, I show you the regulation of insulin release. That means from the beta cell, let's say the insulin is generated and this insulin is ready to be released. That means this insulin is yet not released. Usually insulin are released as vesicle. That means a bag of insulin is getting stored, or it usually stores, a bag of insulin is stored inside the beta cell, and when there's a signal of insulin release, that bag fuses with the cell membrane, and it opens up and everything from that bag gets released in the blood. Now, how this thing is regulated? I mean, what are the signals that regulate your insulin release? Usually, we know that the first thing that comes to your mind when you talk about insulin is, okay, insulin controls the glucose. That means when we have increased glucose in our blood, there is a release of insulin. Another thing is insulin functions to control the glucose also. That means glucose is controlling the insulin release, and also insulin is controlling the glucose regulation. Now, both of both of these things are dependent on each other. Now, here you see the one of the major way or major regulator of the insulin release. That means how the insulin is actually released and what is the signal that helps regulate or helps release the insulin from the beta cell of the pancreas. Here you see in the beta cell of the pancreas, we have this glucose channel that is called the glucose transporter 2. There are several glucose channels we're going to describe. Now, this one is the glucose transporter number 2. And this glucose transporter, through this glucose transporter, glucose is entering this beta cell of the pancreas. Now, the thing is, inside the pancreatic beta cell in the pancreas, there is glycolytic enzyme. And we know when we discussed the biochemistry, I hope you remember, the glucose converts into glucose 6 phosphate, right? That is the first step of the glycolysis. Now, following this step, this glucose to glucose 6 phosphate, following this step, eventually there will be conversion of uh, glucose to pyruvate, and this pyruvate will take part in the Krebs cycle or TCA cycle to generate ATP, right? Also, we discussed the enzyme name that works here, the glucose to glucose 6 phosphate. This conversion requires the enzyme, and that enzyme name is hexokinase. And in this pancreatic beta cell, there's a version of hexokinase works. That enzyme name is not hexokinase, the enzyme name is glucokinase. That means in your pancreatic beta cell, instead of hexokinase, there is glucokinase. And I hope you know that from your knowledge of biochemistry that this hexokinase enzyme has a lower affinity for its substrate. That means unless there's a huge amount of substrate, it is not working that much. Also, we describe it with some mathematical value that is called the Km value. We say that the glucokinase has a higher Km. What does it mean? The Km is a mathematical proportional or mathematical term that describes the affinity of an enzyme to its substrate. That means here the enzyme is glucokinase and the substrate is glucose. That means the glucokinase will work on the glucose and it will add a phosphate with it. That's what the kinase thing does to run it or to proceed into the procedure of glycolysis. That means this glucose will eventually break down and eventually produce pyruvate. Eventually that pyruvate go into your mitochondria to generate energy, right? Now, the thing is that this glucokinase here, it has a higher Km. It means it has less affinity for glucose. Now, what does it mean? It means, or what it makes you think? It makes you think that, okay, to make it work, to make this glucokinase work, I need to have a good amount of glucose. Because it has a lower affinity, it means you need to have a huge number of substrate to push it or to poke the enzyme to work. And that's the beauty of this pancreatic beta cell. Unless there's a huge amount of glucose, this thing, this procedure, this glycolysis will not run that fast or will not run that hugely or extensively. When there is an increased amount of glucose, this procedure of glycolysis will run very smoothly, and that will eventually lead to a huge amount of ATP generation. So that's some discussion from your biochemistry. I'm not going into detail. Just remember that this beta cell, this has this GLUT2 transporter, and this glucose is entering its cell, and this glucose converts into glucose 6-phosphate and follows the steps of the glycolysis. Eventually, the glucose will generate this conversion, or this procedure of glycolysis will generate some ATP. Now, this ATP, we know that ADP eventually converts into ATP in the mitochondrial electron transport chain in the procedure of oxidative phosphorylation. Now, eventually, when there is more ATP, the ATP and ADP ratio will increase, right? If you measure this two ratio, ATP on top, ADP on bottom, if you measure, if there is more ATP, this ratio increases. Or simply, the ATP level rises, right? Now, this ATP, what it does, this ATP causes uh, closing or closure of certain potassium channel. And this potassium channel, when it is ATP sensitive channel, and when this potassium channel is closed, what will happen? Potassium is usually, as you can see, it is going out of this cell, right? Okay, someone says, hey, Jacob, thanks for watching. So when this potassium is a channel is closed, potassium is not going out. Now potassium is a positive charge. Now when this positive charge cannot go out of the cell, they will accumulate in the cell, right? And now eventually this cell becomes more and more and more positive. And when a cell becomes more positive, usually most of the cells of our body, they are in negative charge. But when it becomes positive, we call them that they change their pole of electrical status. That means they were negative, eventually they are now positive. That means they change their pole of electricity. This is called depolarization. That means they were in a pole, now they changed their pole. That's called depolarization. And this thing, this closure of the potassium channel and this accumulation of the potassium inside this beta cell leads to this positivity or this depolarization of this cell. And when this cell is depolarized, when a cell is depolarized, it works as an excitable cell, and it leads to several functioning. Like in this case, the beta cell, when it is depolarized, there's opening of certain voltage gated calcium channel. That means due to that voltage change or due to that electrical status change, the calcium channel opens up. And when the calcium channel opens up, calcium goes in, and we know that calcium is a major regulator of our intracellular mechanisms. Same thing happens here. Calcium comes in, there is increased intracellular calcium. Calcium pushes those already prepared insulin hormone inside the beta cell, and it push this insulin containing vesicles out. Insulin is released. Now, that's how your glucose controls the release of insulin. So again, to recap, when there is high amount of glucose in the blood, glucose comes in through the GLUT2 transporter inside your pancreatic beta cell. Now this you have a if you have increased amount of glucose, that means usually after the meal you have a huge rush of glucose inside your blood. Now that increased amount of glucose will go inside the cell. It will stimulate the glucokinase to work on itself, and this glucokinase will convert the glucose into glucose 6 phosphate. The glycolytic procedure continues. Eventually you will get pyruvate. Pyruvate goes inside the acetyl pyruvate converts into acetyl CoA. Acetyl CoA takes part in Krebs cycle or TCS cycle. It generates several energy producing molecules like NADH, FADH2, and then eventually these molecules take part in oxidative phosphorylation, generates ATP. Now this ATP closes some ATP sensitive potassium channel in the membrane of this beta cell. Those potassium channels are closed. Potassiums cannot go out. Potassiums accumulates. There is more positive charge inside the cell. Eventually this positive charge depolarizes this whole cell. The cell have some voltage difference, and this voltage difference opens up some calcium channel. Calcium comes in, pushes out your insulin containing vesicle. Insulin is out. That's how insulin comes out of your beta cell. Now this is one of the major way insulin is released. There are several other ways the beta cell can be stimulated and it releases insulin, right? Now, here's one way that you can see that glucose is the major regulator of insulin release. Also, insulin release is increased by several, or insulin secretion by the beta cell can be increased by these several procedure. Like one is the glucagon-like peptide. Usually, we know that glucagon it inhibit the secretion of insulin. But this glucagon-like peptide is another type of protein that increases the secretion of insulin by working on the beta cell. Now, one thing you should keep in mind that oral versus IV glucose. That if someone asks you, okay, if you take oral glucose, that means you eat some, let's say, sugar, a donut maybe, or what will be the insulin level in your blood, or in comparison to if someone pushes glucose in your IV, that means in your veins through IV route. Now it means that they're asking that what will be the change in the insulin level if the glucose is taken through mouth or it is directly pushed into your vein. Now, usually when you take the glucose through your mouth, when this glucose is passing down through your GI tract, in different cells of the GI tract, there is a stimulation from this glucose that is passing through the GI tract. And while this food is getting passed through your GI tract, different cells of your GI tract produces some hormones. Those are called incretins. Now, these incretins are glucagon, insulin regulator. As you can see that increased insulin response with oral versus IV glucose. That means if you take the glucose through mouth, there will be more insulin release than if you take it directly into your vein. Because when this food or this glucose is passing down your mouth through your GI tract, that stimulates something called incretins. These incretins are glucagon-like peptide, GIP. GIP is glucose-dependent insulinotropic polypeptide. These are increased, and this regulator, they increases the sensitivity of the beta cell to glucose so that the beta cell releases more and more insulin. So when someone asks you which way you will have more insulin in the blood, you will have more insulin in the blood if you take the food through your mouth rather than you directly push the food into your vein. I mean, you can take the donut through your by your mouth, but how can you push the donut in your vein? Okay, that's a matter of research, I think. All right, so sometimes you know that we take glucose through the vein, but that's an idea. Now, our sympathetic nervous system, it also helps regulate the insulin release. Now, we know that catecholamines work on these different receptors, serotonergic receptor. Now, if the catecholamine works on the alpha-2 receptor on the beta cell, it will lead to decrease in insulin production. And if it works in the beta 2 cell, it increases the insulin release. Now, the thing is, if both receptors are stimulated, now depending on the situation, sometimes the alpha is stimulated, sometimes the beta is stimulated. I think it depends on the level of the catecholamines. That means if you increase the level, there is some change in this receptor selectivity. Now, what will happen if both of the receptors are activated at the same time? Now, usually it is said that alpha 2 activity predominates. That means if norepinephrine is present, it stimulates both alpha 2 and beta 2 receptor. Usually the alpha 2 receptor work or functionality predominates in this case, and that leads to decrease in insulin release. I mean, if norepinephrine is present and it stimulates both alpha 2 and beta 2 receptor on your pancreatic beta cell, it will lead to decrease in insulin release by specifically stimulating the alpha receptor or by specifically running the alpha 2 mediated mechanism. Now, Now, the insulin function, that how the insulin works. You have the insulin now, that how the insulin works. There are several actions of the insulin, right? Usually we say that insulin is an anabolic hormone. That means it's a building block hormone. It likes to build things. Now, how it exactly works. Now, insulin has a receptor. That means insulin is a hormone. When it goes to its target cell, it will bind to some receptor, and that receptor will have some change, conformational change to the receptor, and eventually there is some change in the molecular mechanism inside the cell that leads to the action of the insulin. Like here you can see that insulin binds to its receptor and there is dimerization of the receptor. This is an idea that actually I have this separate discussion of the receptor and intracellular receptor, intracellular signaling mechanism. You can find it in my YouTube channel, USMLE 121. You can search it in the YouTube, or you'll get the link after the session. I'll send you the link. All right, so here you see that insulin, when it binds to the receptor, usually the receptors are two particular molecules. When the insulin comes, they kind of snaps together. This is called receptor dimerization. And when this receptor dimerizes and insulin binds here, there's some signaling in that receptor. And this receptor has some tyrosine residues. Tyrosine is an amino acid. And let's say this receptor has this T attached to it. These tyrosines are here. Now, when this insulin binds to this receptor, there is some kinase activity of this receptor. What does it mean? Kinase is the name of some enzyme, or whenever you see the name kinase, what do you suspect? You suspect that this enzyme works on some substrate. It adds a phosphate group to that substrate with the expenditure of ATP. That means kinase is the name that describes an enzyme that adds a phosphate to its substrate with the expenditure of ATP. Now, same thing is happening here. This receptor has intrinsic tyrosine kinase property. That means this receptor has some enzymatic activity. Whenever they join up together, they run some certain mechanism through which they phosphorylate your tyrosine residues that are attached to this receptor. Now, when this tyrosine phosphorylation happens, this thing eventually runs the cascade of events of the insulin. Like this tyrosine phosphorylation can lead to a generation or activation of several growth factor pathway. This insulin works as usually it's a growth factor. You can say that insulin is a growth factor that works as an anabolic hormone. Anabolic means what? It grows something or it builds something. So you can say that, okay, it's a growth factor pathway. When this tyrosine phosphorylation happens, it can stimulate this pathway, the PI3K pathway, also sometimes called PI3K-AKT pathway, mTOR pathway. I'll discuss it tomorrow, the discussion of several pathways. This phosphoinositide inositide 3 kinase pathway, it stimulates the glycogen, lipid, and protein synthesis. Also, it stimulates some vesicle, and this vesicle has some channel attached to it. That means it's a vesicle, and Surrounding this vesicle, there's some channel. As you can see that these are the channels, and those channels are glucose transporter type 4. And usually this is found in the muscle cell. And you can understand, or it actually makes sense, that let's say uh, there is glucose in the blood. Now this glucose, what it will do, let's say you are taking a meal. When there is increased glucose in the blood, this glucose will go to the pancreatic beta cell. It will stimulate the insulin release. Now, this insulin will circulate and it will work to do its job. Like it will build glycogen. That means it will cluster up the glucose in a more uh, spongy form or less watery form of glucose. The cluster of glucose, we call it glycogen. It may help prevent the lipolysis. That means it will build the lipid. It may help in protein synthesis. It may help your muscle cell to get the glucose. How it works? By this way. In the muscle cell, there is this glucose transporter containing vesicle. That means a vesicle in the muscle cell inside the muscle, and on that vesicle, there are some channels attached to it. Now, whenever insulin is here and insulin stimulates this PI3K AKT-mTOR pathway, it stimulates this vesicle, and this vesicle is kind of pushed towards the membrane. And when it fuses with the membrane, the channel is kind of attached to this membrane. That means if I draw the whole thing, that here is your vesicle and here is the channel. Now this whole vesicle containing the channel, it will go to the membrane and eventually it will fuse up here. This thing will fuse up here. And when it eventually fuses with the membrane, the channel is kind of stuck with this membrane, and it works as a this thing works as a glucose transporter. Your muscle needs glucose, right? Because whenever your muscle is contracting, the ADP is getting utilized, and eventually the muscle contraction is done. You need the ATP to do the contraction again, right? I hope you know that how the muscle contraction works, that the ATP, sorry, the ATP comes and relaxes your muscle. To relax the muscle, you need the ATP and to run the cycle again. Now, this ATP is getting generated from the glucose. Now, your muscle needs the glucose. Then how it gets the glucose? By this pathway, this glucose transporter. How this glucose transporter are translocated, or they are transported through the cell membrane when the insulin comes and activate this PI3K pathway. So insulin goes to the muscle cell, stimulates this pathway so that the muscle cells are tainted by this glucose transporter, so that glucose can enter the muscle cell and help the muscle to contract simply. There are other pathways of the insulin. It's an anabolic hormone, so it will help in cell growth or several other things. There are this pathway, this RAS MAP kinase pathway. It's another cellular pathway through which the insulin leads to cell growth and DNA synthesis. Now, this is the simplest way to describe the insulin synthesis and regulation and the action of the insulin. Now, as we discuss some glucose transporter, let's discuss what are the types of glucose transporter we have. There are some insulin independent glucose transporter, mostly the glucose transporters that are insulin independent. That means they don't need insulin to work. And there is this insulin dependent glucose transporter, that is your GLUT4, that what I just discussed. And it is present in your adipose tissue, it is present in your striated muscle. Now, this striated muscle, whenever it needs glucose, that means whenever we are muscle contracting, let's say you are doing the gym, you are in the gym and you are doing the exercise, and you need the glucose supply inside your muscle cell. And how can you get it? When there is insulin, insulin will push the glucose inside by putting the glucose transporter in the muscle cell. But maybe you were exercising, you haven't eaten something. In that case, usually when you are exercising, the muscle cell are exercised, muscle cell are depolarized, muscle cell, due to the depolarization of the muscle cell, there is release of calcium inside the muscle cell from outside and from inside. The sarcoplasmic reticulum releases calcium. This calcium, this in the striated muscle, this calcium can also push the glucose transporter 4 in the muscle membrane. It means when you are contracting your muscle, there's a chance of glucose transporter 4 expression. But major expression of glucose transporter is done by your insulin. Whenever you have insulin in the blood, insulin goes to your muscle cell. It stimulates these inner machineries that push this glucose transporter containing vesicle in the membrane so that glucose transporter 4 is expressed in your muscle cell. All right. Next is insulin dependent transporter. Insulin independent transporter. That means this transporter don't need insulin to be expressed. Like GLUT1 transporter present in RBCs, brain, cornea, placenta. GLUT2 very important. It's a bidirectional transporter. What does it mean? Bidirectional. It can go both ways, right? Now, how it works. One example can help you understand why it is bidirectional. Let's say this is the small intestine. This is the small intestine, and here is your liver cell. All right. In the liver cell, you have to take the glucose inside the liver cell from your intestine. Now, there's blood vessel in between, right? Because this glucose will be transported from the intestine to the blood and from the blood to the liver, right? Now, here works your glucose 2 transporter. It takes up the glucose from the intestine, and in the liver cell takes up the glucose from the blood. Now, same transporter, it is going to the blood, away from the blood. That means it's kind of bidirectional. It goes to the blood from the intestine, it is taken up from the blood, bidirectional. That's why it is called bidirectional, both ways. It works both ways. To the blood, away from the blood. Now, this same transporter, glucose 2 transporter, we discussed that it stays on your beta cell of the pancreas, where it transports the glucose inside, and where this glucokinase converts the glucose into glucose 6 phosphate, and blah, 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 right? Next thing, GLUT3 present in the brain placenta. In the GLUT5, there is GLUT2. GLUT2 is the transporter. Okay, let me repeat the GLUT2. GLUT2 is the transporter, which is present in this description, this cells, beta cell of the pancreas, liver cell, kidney, small intestine, and now it is said bidirectional. What does it mean? Bidirectional. It can be taken up, it can be used, this GLUT2 transporter is used to take up the glucose from the intestine to the blood. Again, you have the intestine, this glucose needs to go to the blood first, and from the blood it will go to the other cell like pancreatic beta cell or the liver cell. Now this same receptor works to absorb this glucose from the intestine, and also same transporter is used, or it is used, the same transporter is used to absorb the glucose from the blood to the pancreatic beta cell or the liver cell. Now it's kind of bidirectional, right? Because it is going to the blood, it is going away from the blood. That's why this is called a bidirectional transporter. It can go both way, to the blood, away from the blood. Now clear? Is it? That's why it is called bidirectional. I hope that's the confusion, that why it is called bidirectional. Okay. Now, GLUT5 transporter, it also helps transport the fructose, and fructose is necessary for the spermatocyte to survive. And from the GI tract, the fructose uptake is done by this GLUT5. And there's sodium dependent glucose transporter, and that works in our kidney. Some of them works in the small intestine. More to come when we discuss the uh, kidney physiology in the renal system. Now, this discussion actually from the biochemistry, we discussed it earlier, but go over it briefly, that brain needs the glucose for its function, but when there is a starvation, when someone is not getting enough food, the brain can switch its metabolism based on ketone bodies. Now, RBC cannot utilize ketone body because RBC does not have mitochondria. And whenever you have ketone body, you need to have the mitochondria because in the mitochondria, there's certain enzyme, thiophorease. The certain enzyme is there in the mitochondria that is required to convert your ketone bodies into energy producing molecule. But to get energy from the ketone body, you need the thiophorease enzyme. Now, as mitochondria is not present in the RBC, the ketone body cannot be utilized by your RBC. All right. Next are some effects of the insulin. That means it is an anabolic hormone. Usually you see it mostly increases everything. Every level is increased and it is preventing something, some destruction, like it preventing lipolysis. What does it mean? It means it is preventing the breakdown of fat cell. It generates fat cell, big adipose cell it generates, but it prevents the lipolysis. Another thing, other than this anabolic function, it prevents the glucagon release because glucagon is kind of the opposite of insulin. Insulin decreases the glucose level in the blood, glucagon increases the glucose level in the blood. So these are some functions of insulin. You can go for it. Simple. Next is the other hormones, the glucagon. The glucagon is released from the alpha cell of your pancreatic islet, and this thing works almost opposite to your insulin. It causes glycogenolysis. That means glycogen is broken down, and when the glycogen are broken down, it releases glucose. It causes gluconeogenesis. That means more and more glucose are produced. It causes lipolysis and ketone body production. That means the opposite of the insulin. Whatever the insulin does, it does the opposite. Elevates blood sugar level to maintain homeostasis when concentration of bloodstream glucose falls too low. That means when we are fasting, actually, glucagon comes into handy on that case because it increases the glucose level in our body by doing this several procedures. Now, the hypothalamic pituitary hormones, that means that hormones those are generated in the pituitary or generated in the hypothalamus but released in the pituitary, released from the pituitary. So what are these hormones? These different hormones, as you can see, ADH, the antidiuretic hormone. What it does, this antidiuretic hormone, it is produced, first thing you should remember, it is getting produced in the hypothalamus. From the hypothalamus, this production goes into the posterior pituitary, and from the posterior pituitary, it is released. What it does in the kidney, it causes absorption of the solutes, absorption of the electrolyte, because it's an antidiuretic hormone. Now, as it is antidiuretic hormone, what happens that it prevents the diuresis, mostly the water. It reabsorbs the water in the kidney, in the distal tubule of the nephron. It reabsorbs the water so that less water is going out through the urine. That means it's kind of uh, against the diuresis. And that's why this is called antidiuretic hormone. This thing increases stimulus for secretion is its plasma osmolality. What does it mean? If our plasma becomes thicker, plasma becomes more thick, that means less water in the plasma, that thickened plasma or increased osmolality means the plasma is more thick, much more thicker. It is not watery, rather than it's like, let's say it is like the glue. That means it is more thickened form. When it passes through your brain, it stimulates this pituitary gland to secrete the ADH. Now, in case of SIADH, syndrome of inappropriate ADH secretion, there is enough, you can understand that several conditions like pneumonia or some lung cancer, this thing produces ADH inappropriately. And where there can be increased ADH, that might lead to increase in water reabsorption that leads to decrease in plasma osmolality. This plasma becomes much more thinner. TRH, the corticotropin releasing hormone, this thing works released from the hypothalamus works on the anterior pituitary to release these hormones, ACTH, MSH, beta endorphin. And when there is exogenous steroid use, this CRH decreases. Why? Because CRH stimulates the anterior pituitary to release ACTH. ACTH goes to the adrenal gland to produce the steroid hormones. The steroid hormone has a negative feedback over the anterior pituitary and over the hypothalamus to decrease the release of this hormone. Now, if someone is getting exogenous steroids, that means some steroids are pushed inside that person's body from outside, what will happen? That steroid will go into the pituitary and decrease the release of ACTH. Usually, if you have increase in steroid in your body, the ACTH level goes down. It's a negative feedback effect. Now, it's an example that chronic exogenous steroid use decreases the CRH release from the hypothalamus. Okay, someone commented in Spanish, I think. I'm sorry, I don't understand Spanish. Can you write it down in English so I might understand what is your question? Okay, now dopamine, this thing from the anterior pituitary, it would work to decrease the prolactin and decrease the TSH. There are some medication like antipsychotic medications or anti-dopaminergic. So what happened? That dopamine, it brings down the dopamine, this antipsychotic medication. And also dopamine works in our movement by working on the basal ganglia. So that's why when we use the antipsychotic medication, there's chance of movement difficulty, like Parkinson's disease we might get. GHRH, growth hormone releasing hormone, it increases the growth hormone secretion. And there's an analog of growth hormone, GHRH, that is called tesamorelin. This tesamorelin is a GHRH analog. That means they work like the GHRH that stimulate the anterior pituitary to release the growth hormone, and that can be used to treat HIV-associated lipodystrophy. That means in HIV patient, there's a cachexia, loss of protein, loss of lipid, loss of adipose cell from the body. Now you can use this tesamorelin to induce the production of new growths that might correct down that bodily loss. GnRH stimulate the FSH and LH release. GnRH from the hypothalamus releases FSH and LH from the pituitary. And hyperprolactinemia, we discuss it in the next page, that how prolactin stops. Prolactin is the hormone that has a negative feedback over this GnRH. That means it stops the GnRH secretion, and so FSH and LH also goes down. Now, one thing very important to understand and important to remember is that if you have tonic GnRH and if you have pulsatile GnRH, what will happen? If there is tonic GnRH, that means there is continuous GnRH. It's like a faucet or like a tap. Let's say you have a kitchen faucet, and from that faucet, water is running down continuously, right? This is the faucet, and water is running down continuously. This is called the tonic release of GnRH. Now, if there is tonic release of GnRH, that actually suppresses your FSH and LH. But if there is a pulsatile release of GnRH, that means not tonic, tonic means it is continuous, rather than the release of GnRH is pulsatile. That means one drop, it goes down, then stops, another drop, it goes down. That means drop by drop or pulse by pulse. This release of GnRH stimulates the puberty by releasing more FSH and LH. So FSH and LH is controlled by GnRH. When this GnRH is released tonically or continuously, there is less release of FSH and LH. But when there is pulsatile release, that leads to more FSN and LH, that leads to puberty and infertility. Okay, sorry, puberty and fertility, not infertility. MSH released from the anterior pituitary. This thing causes melanogenesis. That means they increases the melanocyte production. Now, this melanogenesis is increased in Cushing disease, as MSH and ACTH share the same precursor molecule, pro-opiomelanocortin. Now, Addison disease also has this hyperpigmentation. Cushing disease also has hyperpigmentation. Now, one type of Cushing disease is called, one type of hypercortisolism is called Cushing disease. Another type of hypercortisolism is called Cushing syndrome. In Cushing syndrome, what happens that there is more cortisol in the blood. That means your adrenal gland is producing more cortisol. But if this increased cortisol level is due to increased production of ACTH from the pituitary, that is called Cushing disease. So what is the difference between Cushing syndrome and disease? Cushing syndrome is your adrenal gland has a pathology, and due to that pathology, it is producing more cortisol. Cushing disease is your pituitary has a pathology and it produces more ACTH, and due to more ACTH, your adrenal gland is producing more cortisol. Both of is hypercortisolism. That means in both cases you have more cortisol, but one is direct pathology of your adrenal gland, another is the pathology of your pituitary gland. Now in Cushing disease, we have more ACTH. And when ACTH is getting generated, there is also generation of MSH, because both ACTH and MSH comes from the same precursor that is called pro-opiomelanocortin. Now, when there is more MSH, this MSH will lead to more melanocyte production, and more melanocyte production means more melanin, and more melanin production by the melanocytes, and more melanin means hyperpigmentation. You get hyperpigmentation in Cushing disease. Also, in Addison's disease, there is increase in ACTH because the adrenal gland is not functioning in Addison disease. So there's no feedback inhibition of the ACTH. ACTH is more, that leads to more MSH production. Don't worry, we'll discuss it again in respective section of the pathology. Oxytocin. Oxytocin, it causes uterine contraction during labor, and also it causes a milk letdown reflex. When the baby sucks the mother's nipple, there's some stimulation that leads to release of oxytocin that eventually leads to the milk release from this mammary glands. Prolactin, it decreases the GnRH. Prolactin can be increased in pituitary adenoma, that means tumor of the pituitary gland. If it is prolactin producing tumor, that leads to increased prolactin level in the blood, that leads to amenorrhea, because prolactin opposes your GnRH, gonadotropin releasing hormone, which eventually oppose the FSH and LH release, and there will be amenorrhea. And as FSH and LH is not released, estrogen and progesterone will not be released, and estrogen is a major regulator of bone formation in the female body. There will be osteoporosis, hypogonadism, galactorrhea in male. Somatostatin is a inhibitor of the growth hormone and also TSH, that means thyroid stimulating hormone. These are analogs used to treat acromegaly. Thyrotropin releasing hormone, it increases the TSH and prolactin secretion. If there is increase in TRH, thyrotropin releasing hormone, it may cause prolactin secretion that might lead to galactorrhea. That means if someone has hypothyroidism, usually in hypothyroid patient, what happens? The TSH increases because there is no negative feedback. So the TSH level increases. TSH thinks, okay, there is no thyroid hormone, let's increase so that I can produce more thyroid hormone. Now, this same thing, this hypothyroidism, can stimulate the hypothalamus to generate more TRH. And that increased TRH, as it has action over the prolactin, it can stimulate the prolactin release, and this prolactin secretion eventually may lead to galactorrhea in male. Now, here you see that the mechanism of prolactin and how it is regulated. The prolactin is a hormone that is getting released from the anterior pituitary. This prolactin has some stimulus, some inhibitor. Like prolactin, first, what the prolactin does, prolactin on the breast, as you can see, it is stimulating the milk production, right? Now, if it is inappropriate, it can cause galactorrhea. That means hyperprolactinemia can cause galactorrhea in male, in female, both actually. And this prolactin can stimulate inhibit the GnRH, the gonadotropin releasing hormone. And when the GnRH is inhibited, it will eventually lead to less FSH and less LH. And when the FSH and LH is less, ovulation and spermatogenesis will be stopped. That leads to amenorrhea or infertility in amenorrhea in female, infertility in male. Now, how this prolactin is regulated? One thing you see that TRH stimulate the prolactin release, and TRH itself is inhibited, or it has a negative feedback from increased plasma T3, T4. That means thyroid hormone, if the thyroid hormone increases in the body, in the blood, it inhibits the TRH release. Now, if there is hypothyroidism, this negative feedback will not work, so the TRH will be there, and this TRH will stimulate this anterior pituitary to release prolactin. Hypothalamus has control over this dopamine and TRH. Hypothalamus has the effect on the release of dopamine, and this dopamine can work on the anterior pituitary to inhibit the prolactin. That means dopamine is a inhibitor of prolactin release. Now, the thing is, if there is sight or cry of the baby, when a mother sees a baby or when a mother hears the cry of the baby, what happens that in the higher cortical center of the mother's brain, it inhibits this part of the hypothalamus that controls the dopamine. Now, when this part of hypothalamus is not working, the dopamine cannot work. And if dopamine is not working, it cannot inhibit the prolactin release. And prolactin will be released and it will stimulate the breast to be prepared to give milk to the baby. How amazing it can be. Now, another thing is prolactin has stimulatory effect on its inhibitory pathway. That means prolactin itself has a negative effect on its pathway because prolactin itself can stimulate the hypothalamic dopamine pathway that leads to inhibition of prolactin release. Another thing is you see that dopamine antagonists like bromocriptine or chest wall injury via autonomic nervous system or nipple stimulation. These processes can inhibit the dopamine directly, and when the dopamine is not working, that leads to more prolactin release because dopamine is not there, so there can be release of more prolactin because there is no inhibitory function over the prolactin. Last thing is if someone has renal failure, the kidney is not functioning, the prolactin will accumulate in the body because prolactin, when it is done working, it usually with the blood goes to the kidney, it filters out through the urine. So if there's renal failure, there'll be less excretion of prolactin, more accumulation of prolactin that leads to increased level of prolactin. Another thing is estrogen, which is more in pregnancy. It stimulates the prolactin production and release, but during pregnancy, we don't see the milking action. And usually, why it is happening? Because in pregnancy there is more progesterone, and this progesterone is inhibiting the prolactin from working on the breast. And that's why, although the prolactin is getting produced by the action of estrogen, it cannot work on the breast during pregnancy because there is progesterone also. Whenever the placenta is removed after the delivery, this progesterone function is gone. Now the prolactin can easily work in the breast that leads to milk secretion. So This is about the prolactin, very briefly, very shortly, about the physiology of the prolactin. All right. I think that's it. That's for the first discussion. We'll have continue this discussion on endocrine system, the USMLE step 1 endocrine system. Up to the next 4 days, we'll discuss the rest of the physiology. That means the very important, the adrenal gland physiology, how these different hormones are generated in the adrenal gland. We'll discuss the pathology. We'll discuss much more interesting pathology like diabetic ketoacidosis and this type of things. And we'll discuss the very important pharmacology section in these 4 days. So if you want to join in, just let me know. Again, here is the email address at the very beginning of the session that I showed you. That this is the email address, drstutor@gmail.com. Let me know if you want to join in. And also, you see in the if you're joining through the Facebook, there's a in the comment section, there's a link from the page USMLE 1-to-1, the page I run, a Google form. You can fill out that form to as a feedback form, and those who are joining through the Zoom, you will also get the feedback form. But if you are my current student, don't fill it out. This is for you if you are joining for the first time this type of session. All right, so that's it. Do you have any question? Anyone from the Zoom or from the Facebook have any question? All right, so let me know if you have any question. You can send me email. And thanks for joining. And those who are joining through the uh, Zoom webinar, I'll send you this handout, this note. And if you're interested, if you're participating through the Facebook, just send me an email to this email address. If you want a handout, I'll send you that handout. And also, this session will be available in the Facebook as a recorded live session, and also will be available in my YouTube channel, USMLE 1-to-1. Thanks for watching. Thanks for joining. Take care, everyone. See you tomorrow. Goodbye.